Hello everyone, this is Dr. Yasin from Abeda Inamdar Senior College, Department of Chemistry, Assistant Professor. I am MSc, B.Ed, NED, MPhil, PhD. Now, today I am going to discuss about the very new syllabus of our Abeda Inamdar Senior College, that is has to be autonomous. Now I am going to give you the lecture on biomolecules. So before that, we should know about the syllabus of the biomolecules. So it is considered as we have a choice-based credit system which can be followed by 2019 pattern in T.Y.B.Sc chemistry. This is the one of your subject, that is the chemistry of biomolecules, which is having credit of 2. That's almost having the 36 lecture. It is having a code D.S.E.C. You can have one more option if you want to choose, that is the material science. So I'm going to take the chemistry of biomolecules. So what we have to do in the whole of first semester of the T.Y.B.Sc chemistry course. So there are the title and the topic of the each chapter has given in front of you. So there will be a total number of chapter is 6 chapters. The very first is Introduction to Molecular Logic of Life, which is contained number of lecture is 3 for that. The second is most important in the biomolecules, that is carbohydrates, which is having the 7 number of lectures. Third number of the chemistry of biomolecules are lipids, and then fourth is which is having lipids is having the lecture 6 for this. Amino acids and proteins having 8 lectures, enzymes having almost 6 lectures, and most important the hormones, which consist of 6 lectures as well. So total number of lecture will be 36. So I am going to give you the brief idea about the syllabus. Each chapter contains there are different variation we have to study. So the very first chapter, that is Introduction to Molecular Logic Life, which is unicellular, multicellular organisms, prokaryotics and eukaryotics, list of cell organelles and its functions, molecules that constitutes the organization of cell and its organelles, types of bonds in biomolecules. So this we have to learn about the what are the molecular logic life, and this is the introductory part which consists of 3 lectures. The second most important introduction of the biochemistry of biomolecules will be a carbohydrates. We'll go into detail of their introduction, the classification of the carbohydrates, then their structure and the biological significance. We have to read, we have to learn about the what are the concept of the anomers, epimers, reducing and the non-reducing sugars, the mutarotations, inversions as well. Then we have to learn about in the 7 lectures the reaction of the glucose with acid, base, phenylhydrazine, oxidizing agents, reducing agents, and its significance as well. And as well as we have to learn about in the carbohydrate, that is the glycosidic bonds, and that glycosidic bonds will be covered our carbohydrate chapters. Then next we will learn about the chapter lipids, which consists of 6 lectures. In this we have to have the introduction part, classification of lipids, their structures and the biological significance, the different reaction we have to learn, that is the saponification reaction, hydrolysis, emulsification, oxidization. So concept of the saponification number we have to calculate the acid number, iodine number, and their significance. That all this part we have to learn in the lipids. The next one is the rancidity and the types of the lipoproteins and their significance, which consists of what blood groups of substance will contain in the lipids. That we have to learn in this chapter. The next chapter will be the amino acids and the proteins. The amino acid and proteins, we have to learn about their classification of amino acids, the different concept of the amylophosphates, isoelectric pH, Reuter ions, titration curve of glycine, that we have to learn in this amino acid classification. As well as the classification, we have to learn about the reaction of the amino acids with the ninhydrine, sanjardanyl chloride, diabesyl chloride, and the adamants reagents and their significance as well. We have to learn about in this chapter the peptide bonds and its features, how it will be, it will be reacting with the different factors of this. The proteins will be the next classification based on the function and the nutrition and its composition. The organizational and the structural proteins, primary, secondary, and tertiary, will have the Cartridge structures. We have to learn all the structure of the proteins as well. So enzymes are the next chapter that we have to learn, as well as the classification. We have to learn its active sites of the enzymes, ES complex formation. We have to see enzyme specificity, factors affecting enzymes activity as well, and the basic enzymes kinetics, because the and LB equations also we have to learn in this, which are having the significance of Km. There are the different types of enzymes, which inhibitions, concept of conjugated enzymes as well, the prostatic groups, epinozymes, holonozymes, and the coenzymes of vitamins in the industrial application as well. We have to see there are the ample of applications of the enzymes. So we have to learn and we have to contribute the chemistry of biomolecules with the help of enzymes also. So the next chapter and the last chapter will be the hormones. There are the introduction of the endocrine glands and their hormones, biochemical nature of hormones, as well as the mechanism of the action of lipophilic and the hydrophilic hormones. So here are the 6 lecture for the hormones, and we will finish it with this semester. For what for this purpose, what we have to refer the different types of references books they are given. The Leibniz principle of biochemistry by Nelson, you have to see the edition. The latest edition will be the 4th. The biochemistry by the Satyanarayan and the Harper illustrated biochemistry. This will give you the reference books, the very huge idea about the biomolecules of the chemistry of biomolecules. There are the M number of the books are there. We can see for the different reaction and the mechanism. Morrison Boyd, this is the very basic book for the introduction of chemistry. Biophysical techniques by Upadhyay and Nath. These are the 3rd revised edition we have mentioned over here. The volume, that is FINAR and FINAR 2, volume FINAR and FINAR volume 1 and 2 respectively. You have to see for the mechanism, reactions, and the chemistry of biomolecules for the same. So what are the learning objective of this syllabus and the chapter? So the learning objective of this content chemistry of biomolecules will be why to learn about the logic, introduction of the molecular logic of the life. Molecular logic of the life will be the student can understand the cell types, different cell of the types, different bacterial cell, plant cell, as well as the animal cell as well, different composition as well as biological composition, organization of the cell membrane. We have to learn in the chemistry of biomolecules, their structure, the function, the various cells, organelles of plant and animal cells. There are the concept of the biomolecules as well. So bonds that link to monomeric units to form the macromolecules. So these are the very important objectives and outcomes from the learning from this chapter as well. So we have to go for the next. Why we have to learn the carbohydrates? The student will understand the types of the carbohydrates and their biochemical significance, because the carbohydrates having the lot of significance in living organisms. There are structure of the carbohydrates we have to learn, and the reaction of the carbohydrates with the glucose as example, and properties of the different carbohydrates as we have learned in the 12th standard and as well. So we have to have the lipids. Why lipids we have to learn? The students needs to know about the types of lipids, their examples, and the different structure and the properties of the lipids as well. Amino acids and the proteins. In this, the students can understand the structure of the types of amino acids, the reaction of amino acids, the properties of the amino acids, and the bond formation which can be done with the peptide bond formation. Types of proteins we have to learn in this, because students can understand this structural features in the proteins, effect of pH on structure of amino acids, what are the different effects of the pH. So that had to be done in this objective of the for the students to learn of this determination of nitrogen and carbon terminus of the peptide chain as well. Why we have to learn enzymes? So students can learn the classes of enzyme with the substances, subclasses, and the examples, which has been specificity of the equation of enzyme kinetics, that is its significance. Also they have to learn a feature of the various types of enzymes, inhibition and industrial application. There are the ample of applications that I have already told you about the enzymes as well. This is all about the syllabus of the SPPU chemistry and chemistry of biomolecules. So we can learn in the next lecture. We can start with the introduction of the molecular biology life.